we will go through the effect of exercise on blood glucose level. So, effect of exercise on blood glucose level in a human subject is our today's topic. Along with observing the effect of exercise, second thing that we will observe is about this small instrument called glucometer. Glucometer. Gluco from glucose, meter measuring. So as it is used for measurement of glucose level, so it is called glucometer. So today we will also see that how this glucometer works and how we can use it to determine glucose within few minutes, just with a small drop or a single drop of blood. So that's why this instrument is now very common with the diabetics. So two parts. First part, let's see the effect of exercise on blood glucose level. Exercise or physical activity, it involves more and more muscular activity. Whenever we say we are exercising, we are performing some physical activity, it means we are using more and more our skeletal muscles. These skeletal muscles, when they are in use, they need energy. And this energy is supplied, this energy is provided to these muscles by the glucose. So it means when we want to perform some physical activity, our muscles need a continuous supply of energy through glucose. This glucose, which is required for the exercising muscles, it is provided, number one, by increasing the blood supply to the exercising muscles. So whenever the muscles starts activity, the blood flow to that particular area increases. And this increased blood supply, increased blood flow, it provides more and more glucose, more and more energy substances to be utilized by the muscles. Number two, second source of glucose for these exercising muscles is by breaking the stored glycogen. The glycogen which is kept stored in the muscles, it is broken down and the released glucose is utilized by the muscles. Thirdly, in order to supply more and more demand of the glucose, the process of gluconeogenesis starts. Means synthesizing the glucose from sources other than carbohydrate. And finally, the other substances like fats are utilized by the muscles. Now this increased utility of glucose is one part. But you know that there are two ways to utilize glucose, aerobically and anaerobically. So initially the exercising muscles, they will use glucose in the presence of oxygen or aerobically, so that more and more energy can be obtained, and also the glucose will be broken down till the last product, that is carbon dioxide and water. But this demand of increased oxygen is initially completed by increased activity of the respiratory system and increased supply of blood by the cardiovascular system. But later on, as the respiratory muscles also gets exhausted, fatigue, and therefore no further increase in oxygen supply can take place, and therefore the muscles started using glucose anaerobically. This anaerobic use of glucose, it results not only in less supply of energy, but also accumulation of lactic acid. Anaerobically, when further increase in oxygen cannot be made. The respiratory system is working at maximum, no further increase. This accumulation of the lactic acid, it will result in the development of pain and a change of pH to acidic side, and we say that our muscles gets fatigued. And this pain and other changes, they force us to stop our exercise. It means that the basic change during exercise is a more and more utilization of glucose. And therefore, the blood glucose level will fall.
Lose.
So it means that the effect of exercise on blood glucose level is to reduce it, a hypoglycemic effect. And this is the reason why we ask the people to do exercise, so that more and more glucose will move inside us. Along with these changes, there are certain other changes also that will take place in the muscles themselves. When the exercise is performed at regular basis, not occasionally, but on regular basis, like 3 times a week, 4 times a week, then this will result in number one, increase in the muscle mass. How increase in muscle mass? Both by increase in the number of the muscle cells, and number two, by the increase in the size of the muscle cells. So both the size of already present muscle cell will increase, they will become larger, as well as new muscle cells will be formed. and as such the muscle mass will be increased, which is known as hypertrophy of the muscles. Muscular hypertrophy. Number 2, more and more new capillaries will be developed in the muscle in order to supply the increasingly required blood. To supply more and more blood to the exercising muscles, the number of new capillaries get more and more. Number 3, the number of mitochondria in the exercising muscles, it will increase in order to supply more and more energy. And number 4, the amount or the quantity of myoglobin will increase. Myoglobin is a red color pigment just like hemoglobin found in the muscles that can also combine with
Oxygen.
but present in the muscles, so called myoglobin. Number 2, increase in number of capillaries. So increase in muscle mass, increase in number of capillaries, increase in number of mitochondria, increase in the amount of myoglobin. These are all the changes that will take place in the exercising muscles, as well as increase in the amount of stored glycogen, muscle glycogen. So all these changes, they will take place in the exercising muscles and produce a beneficial effect and advantageous effect on our body and on our health. Coming to the point that how we will measure this glucose level before and after exercise. There are various methods to measure the glucose level. Like before, we measure glucose by using spectrophotometer. But one of a very simple method is to measure glucose by using glucometer.
في عندك برنامج هنا لما تكتب كده ينقل خطك ما ركب الورقة والقلم.
ترى معفن هذا خطك اذا خطك مضبوط
So one of the convenient and easy method to determine the glucose is by glucometer. Why this glucometer? Why this glucometer is, we say, so convenient and important? The reason is that with the start of use of this glucometer, the measurement of blood glucose level become the easiest way for particularly the diabetics. Because diabetics, in many cases, particularly those who are on the insulin, they have to measure their glucose every time before injecting insulin. And in that case, it was very difficult for them to every time to go to the lab and get their glucose checked. But now with the availability of this glucometer, it becomes very simple for them. They just check their glucose by themselves and take their insulin. So this is very safe, very convenient, and very simple, very easy method. This glucometer, it has some parts. Like the first part is a screen in order to show the different values. Value of glucose, number of the coding chip, level of the glucose, value of the glucose, all these are represented on the screen. Number two, a second part is here, a small space or cavity or opening. This space or cavity or opening is for the introduction of a small strip. It is called a strip, S-T-R-I-P, strip. And as it is used for glucose, therefore it is called glucose strip. So this small space, it is for the introduction of this glucose strip inside. Okay? Thirdly, on the back side of this glucometer, there is a space available for the attachment of this battery. For the attachment of battery. So these three spaces. Number four, on the side of the glucometer, here also on the side, there is a space available, and in this space a small coding chip. It is called coding chip. A coding chip has to be introduced here like this. This coding chip, it is available whenever you will purchase the box of glucose strips. With every glucose strips box, a specific coding chip having information about these glucose strips is available here in the bottle. So you have to open the bottle, take out the coding chip, put it inside here, and then you can introduce your glucose strips. So with every box of the glucose strips, a coding chip is also available, and on the box, the number of the coding chip will be provided. Like here it is written 385. So 385 is the number of the coding chip, which is also given here on the box. And whenever you will start the glucometer, first thing that appears on the screen will be the number of the coding chip, 385. After these things, now let's have few things about the strip. The glucose strip, it has two sides. One side is here, you can see that a black area is there, and below there is a circular yellow colored area. This is one side. Second side is here, where totally white except a small colored square area. Now which side is the active side? This second one. The total white with a small colored area. The color area may be orange, the color area may be green, the color area may be yellow, any color. But here in this case it is orange. So this white with colored square area, it is the active site, and the colored area indicates the place where you have to put the drop of the blood. Where you have to put the drop of the blood. Okay? Number 2, when you are inserting this strip into the glucometer, this colored area has to be put upside, not downside. If downside, it will be wrong. It should be upside. Number 2, which area should be inside the glucometer? This one or this one? For that purpose, an arrow is given. Arrow. This is called arrow. So in the direction of the arrow, you have to introduce it into the glucometer. Otherwise, if you introduce it in the wrong direction, it will not work. So for proper working, correct direction of the glucose strip is required. Okay? The second thing is that when you are putting the drop of blood here on the colored area, what is the use of this circular yellow area on the back side? What is its use?
Lights.
What will happen that when you put the drop of blood here on the colored area, a chemical reaction take place and the screen will give you the value of glucose. As a result of this chemical reaction, the backside yellow circular area, its color will be changed from yellow to green, dark green, reddish, orangish, any color, depending upon glucose level. Now what is the advantage? That suppose that one day your battery is out of order or something wrong with your glucometer and you cannot check your glucose level. But in order to take insulin, it is must. So what you can do, just you take a drop of blood, put it on the colored area as usual. When the reaction takes place and the color change here on the backside area, now what you can do, take this glucose strips box. Here you can see different colors are given. Now this circular area color, you can match that this color matches with this one or this one or this one. Wherever it matches, you can read how much glucose level is given here. And in this way, by using this circular area, you can find out a rough value of your glucose. Not accurate, but at least a rough idea about the glucose. And you can take your insulin. So that's why the advantage of this back colorful circular area is that the color that changes here, you can match it with the colors given on the box, and by that you can determine the glucose level. But this is not very accurate, as the glucometer, it will give you only a rough idea about glucose. Now coming to the another point, that for the purpose of glucometer, we need only a single drop. Therefore, usually we use capillary blood. Just with a prick, the capillary will puncture and take out the blood. So a capillary blood is usually used, but arterial or venous blood can also be used, no problem in it, but usually capillary blood is used. While you are using this glucose strip and glucometer, the drop of blood should be of such a size that the whole area of this whole colored area should be properly covered with blood. Otherwise, if the drop of blood is so small that only half the colored area is covered or less than half is covered, then the glucometer will give you either a wrong value or it will give you a letter E on the screen, like this, letter E. When the letter E appears on the screen, it means error. E for error. Error yani?
Wrong.
So, wrong. So, error appears means E appear on the screen. And whenever there is something wrong in your procedure, the letter E will appear on the screen, and it means that your procedure is wrong, something wrong in it, and you cannot get the glucose value. So that's why if you will not cover the colored area properly with the blood, letter E will appear on the screen and you will not get the proper result. Okay? Now, last point about it, how to use it. In order to use it, what you do, then you take the glucometer, you take your glucose strip and you insert the strip here. The glucometer will start. First, 701 is the coding chip number, it comes, and now you can see a drop of blood starts sparking, flushing on the screen. When the drop of blood starts flushing on the screen, it means the meter is ready now. And so you have to quickly clean your finger first, then prick it, press it, make a big drop, and then put the big drop on this lighting colored area. You can see the colored area is lighting as well. So on this lighting colored area, put the blood drop. Within few seconds, the value will appear on the screen. But if you first take the strip, put the drop of blood here, and now you are trying to insert it in the glucometer, it will give you a big letter E. So don't put the blood drop as usually the students do in the exam, that they put the blood drop first here and then now they are trying to put it inside the glucometer. Until and unless that first you introduce it, let the meter ready and then put the drop of blood, only then the value will come. Okay? Clear? Yes.
كل واحد ياخذ شريط ولا نفس الساعة؟ ها؟
Okay. Now who will be the volunteer today? Not only for checking his so let's see. Usually the
taking sugar and راسك.
العزيز راسك وسع.
Okay, now you see that before exercise it is 522 milligram. Very high level. Huh? Now let's see after exercise how much glucose level comes back.
معدومة.
What you have
ما شاء الله ما شاء الله ما شاء الله 522 مريض ما شاء الله تبارك الرحمن ما تعبت يا مفاز لا انا ما عندي فرح انا ما عندي اسرع الحيدر عمي يقول بعد نجيبها بسرعه بس 6 ثواني انت كلنا
Last time I did this with group 2 in the first semester. Exercise tolerance test. Hardly 1 or 2 students complete 3 minutes. Remaining is for nobody. Somebody 1 minute, somebody 2 minutes.
ما شاء الله تبارك الله.
1 or 2 students. Even
والله الحين تحس شجعوه يا اخي مو
اي والله افضل
يا خلاص يقول ما يحتاج تسمع احط ستوب ما عندي
صوت ثاني نسوي تكرار بيحاول يصيد خلاص دوبك دقيقة ان شاء الله في الكاميرا والله جدا سمعت صوت انا حافظ لكن ليش بس غلطة قهرني والله
ما ادري اكثر لكن انا مني ما حسيتها زي بس حسيت بس بشتغل
على نفسها تعيد
يلا قصة الستة بدون
How much was before? 522. 522. 522.
والله اقول لك زاد ما شاء الله
المفروض يتحكم في السكر حقه
تعال نطوع اقرا عليك نقرا عليك
Now suppose you write this result that 522 before exercise and 542 after exercise. And the examiner will ask you that okay, the effect of exercise is to reduce the glucose level, but here in this case the glucose level is increased, not decreased. So in your opinion, what may be the possible reason? The first thing that is coming in your mind is that maybe some problem in the instrument. But even if something is problem in the instrument,
should be reduced.
then why this problem is after the exercise only? Why not before the exercise? The instrument is giving value. You can say that if the instrument is not working properly, it means the 522 value was wrong. Maybe it is 300, not 500. But after exercise, if it is decreasing, why it is giving not decrease? So it means that one possibility is not in the instrument. The possibility may be that the exercise that we perform
Not enough.
is not enough.
Not enough.
Because when we say that the exercise decreases the glucose level, it doesn't mean that just 1 minute or 2 minute exercise will decrease the glucose level.
Half an hour.
At least the exercise should be in such a way that it will make a significant change in the respiratory and the heart rate. Sweating starts. Then you can say that this will burn out the glucose. So here in our case, just asking him to do only for 2 minutes and then we are checking. But why it is increased? Okay, not enough exercise, it should not it decrease, but at least the value should remain same. 522, 523, 525. Why 545?
Why did it change?
The reason is
Gluconeogenesis.
that whenever you will perform some activity, your sympathetic nervous system will be stimulated, leading to the release of adrenaline, noradrenaline. And adrenaline, noradrenaline make our body able to fight against any stressful conditions by making more and more glucose available, increasing the blood glucose level. So it is the stress of the exercise that increases the glucose level here. But if we continue the exercise at least for half an hour, then naturally the level will
Glycogen.
Okay? So in your sheet, when you write this result, that initially before exercise 522, after exercise 545, this all explanation you should write there. Okay? Clear? Everybody now knows how to use this glucometer. Can use in the exam.